From our chapter on blood, we know that the master stem cell creating all of our white blood cells, red blood cells, is called a hematopoietic stem cell. This stem cell is capable of dividing into two other quasi-stem cells. I mean, they're still stem cells, but they're not as able to differentiate in as many things as a hematopoietic stem cell. These two other stem cells, myeloid stem cells and lymphoid stem cells, can then differentiate into the final cells that we see in tissue and blood. We have before discussed this myeloid stem cell, which gives rise to erythrocytes, platelets, and 4 out of the 5 leukocytes. We've talked about macrophages, cells that eat debris in other cells. I have mentioned dendritic cells, which are cells that drink fluids in order to ingest debris or other cells. They're called pinocytes, not phagocytes. Both macrophages and dendritic cells, they derive from monocytes that are floating around in the blood and decide to get out and take residence in a certain tissue. What we haven't previously discussed is a lymphoid stem cell, which is capable of dividing into T cells, B cells, and other cells that are called natural killer cells that we abbreviate NK cells. T cells can divide into a whole bunch more versions of T cells, and B cells can divide into plasma cells that then create antibodies. Monocytes are white blood cells that derive from the myeloid stem cell. Monocytes float around in your blood, however, they don't spend the entirety of their life in blood circulation. Monocytes will move into tissues in the body and they take a permanent residence in those tissues. Once there, we usually give the monocyte a more specific name according to the tissue. You might not have realized this, but osteoclasts, which were cells that were capable of breaking down bone, they were not derived from the stem cell that the other bone cells were derived from. Osteoclasts are actually a monocyte that has moved into bone to take a permanent residence, and it phagocytoses bone. In the brain, we have microglia. In the liver, we call our monocytes Kupffer cells, and in your lungs, someone has applied the ingenious name of alveolar macrophages. I'm kidding, I'm sorry. That's where they live, in the alveoli. There are tons of different macrophages in the spleen, not just the 4 that I have listed here. You have leukocytes floating around in your blood, but that's not exactly where they do their work. There are macrophages, which are eosinophils and neutrophils. And over here on the right side of this diagram, I have a blood vessel with a neutrophil floating around in it. Let's say that your skin becomes wounded, like it is here in this picture. And whatever punctured your skin is covered in tiny little pill-shaped bacteria, which are called bacilli. The first thing that happens is that a resident macrophage that lives in your dermis kind of goes over and swallows up one of those bacteria. And the macrophage releases all of these chemicals called cytokines. And there are also interleukins. And these cytokines and interleukins, they diffuse away from the macrophage and they enter into nearby capillaries in your dermis, which are the subpapillary or the cutaneous plexuses of the dermis. Once inside the endothelium of the capillary, the cytokines are displayed on little receptors sticking off the endothelial cells. And these little receptors are kind of like hanging out in the blood vessel, waiting for white blood cells to bind with them. So white blood cells, they're just flowing by and in the blood, and they sense this signal of the cytokine or interleukin. And the white blood cell, it starts to roll along the outside of the epithelium, hoping that it'll be able to attach to one of those receptors that it sensed with the cytokine that's being displayed. When the attachment happens, the endothelial cells of the blood vessel, they start to dilate. And this allows enough space for that white blood cell there to fit between these endothelial cells and squeeze itself into the tissue. Now, the white blood cell is drawn toward the injury site and performs something called chemotaxis, which is just the movement toward a specific stimulus, kind of like me and how I go toward coffee. White blood cells are also capable of something called diapedesis, which is where it's like a moving process or a walking process, wherein they can disassemble some of their cytoplasm to like move forward through the tissue. Once they get to the injury site, the neutrophil performs phagocytosis to consume the pathogen, and then they start to release their cytokines and interleukins and recruit more and more white blood cells to come to the area. And this is a positive feedback mechanism, as one neutrophil can recruit so many other neutrophils to come to this area. Natural killer cells do not require to know what it is they are killing. And it's this anonymity that makes them a part of the innate immune system, even though they're lymphocytes. Natural killer cells they're like always frisking the cells of your body and like trying to confirm that the cells are in fact your cells, right? And that they're wearing your identification tag. If a natural killer cell senses that another cell is in any way invaded by a virus or possibly is a pathogen itself, a natural killer cell, it'll just kill whatever it finds without regard for its identity. A natural killer cell does this by attaching itself to the target cell membrane. And here's our natural killer cell, and here's our target cell. And the first thing it does is that it secretes an enzyme called perforin. And perforin then assembles itself on the cell membrane of the pathogen or the faulty cell, and it makes a hole in the cell. That's what this is right here. It's basically a hole, like a tunnel into the cell. And it's at this point that the cell could possibly die from its own components, like spewing out of the hole. But if that doesn't happen, the natural killer cell is not going to like wait for that to happen. What it does is the natural killer cell secretes granzymes into the target cell, ensuring that it will die. And these granzymes, they act like little digestive stomachs, and they basically digest the target cell from the inside out. And so now here is our dead target cell. And so the natural killer cell, it just kind of like moves on to the next cell in line, and it lets that dead target cell hang out for some kind of macrophage to come and then consume this invader. We're going to find out that in adaptive identity, it's these macrophages that will then display pieces, just pieces, of the invading cell on receptors on its plasma membrane, kind of like saying, hey, I found this. I don't think it's supposed to be here. Hey,